So how DNA was found out or discovered to be completed or replicated through a semi-conservative mechanism was done by Meselson and Stahl. So back then, in around the 1950s, there were three types of replications that were thought of that DNA would go through. So the first is semi-conservative replication. So you start with a normal DNA strand, and when it duplicates, if it's semi-conservative, then half of the strand, or half of one strand, will be old DNA and half of the other strand will be new DNA. So as a result, you get two strands that are half new and half old. In comparison to that, we have conservative replication. And conservative replication is when you take one whole DNA strand, you replicate it, and it does not split. So you have one completely old DNA strand and you have one completely new DNA strand. That's why it's conservative. You're conserving one entire DNA. And the last one that was not too popular was dispersive replication. So dispersive replication is where DNA is replicated randomly, or little parts of DNA are replicated. So you start off with one entire DNA strand, and as it replicates, you have some parts that are old DNA, you have some parts that are new DNA, and then old DNA again. So it's completely random. So how Meselson and Stahl studied that it was semi-conservative was they took E. coli DNA and they grew it into heavy nitrogen. So heavy nitrogen is important because it's how they're able to see what replication was occurring. So when they grew it into heavy nitrogen, or nitrogen 15, the DNA from the E. coli just continued to replicate, and it replicated into, for example, let's say 4 strands. Then at T equals 0, or starting the experiment, they moved it to a light nitrogen. The light nitrogen was N14, and N14 is different in this case, or in the drawing, from heavy nitrogen by a lighter color. So heavy nitrogen will be dark black, whereas light nitrogen will be gray. So when it's replicated inside the light nitrogen, what happened is DNA split and then the new light nitrogen bonded onto it. So what happens is you have two strands of DNA from one, and it's a mix. Half of it is old, or heavy nitrogen, and the other half is light nitrogen. Then they analyzed the weight of it, or the density of it, through centrifugation. But before they actually studied the results, they allowed it for one more replication. So when you took the mix and replicated it inside the light nitrogen, the result was that they had two strands that were mixed and two strands that were light. So after 2 cycles, you have 50% mix and 50% light. So how did they visualize this? And they visualized it in centrifugation. So after they centrifuged it, they looked at the density. So positive, negative, and when at tri equals 0, or T equals 0, you have an entirely heavy strand, or an entirely N15. So it's shaded in completely black and there's no other lines. But after 1 cycle, you had a mix between heavy and light DNA. So it's in between for the weight of nitrogen 15 and nitrogen 14 because it's a mix. And it ended up being a little bit higher. But since it's 100% mix, then they only saw 1 bar. After a second trial, they had 50% mix and 50% light nitrogen. So if you look here, the result shows 2 bars where 50% are nitrogen 14 and 50% are mix. And if they replicated it a third time, what would happen is you have 75% light nitrogen and 25% heavy nitrogen, or mixed nitrogen actually. So as you can see, this is a semi-conservative replication because there is a mixed DNA. If it was conservative replication, then what would result is you have 50% heavy, 50% light, but that wasn't the case. They had 50% mix and 50% light. if it was, they didn't study for dispersive replication for this, so the entire experiment, Meselson and Stahl, only revealed that DNA replication was not conservative, which means that it was semi-conservative.